Okay. I should be done grading the exam, the essay portion of the exam by the end of today, and hopefully I can give you feedback on. I'll make an announcement, and then I'll probably say for Wednesday it's a good idea to bring your laptop so that we can go over the exam in person, and I can talk to you more about it during Wednesday's lecture. So kind of plan on that. Make sure you have your laptop if you want to see your exam. Okay. And then I won't go through every single question, but I'll kind of hit the highlights and then let you look at stuff. And then if you want to go over it in more detail, please set up an appointment with me so that we can talk specifically about your exam and how it went and ways to improve if you're worried. Okay, I have strategies that work. So, but right now we need to get into the continue on with the mycorrhizal fungi and how important they are for so many plants because they help the plant get access to minerals and water that they themselves can't physically access. And a lot of it has to do with just the size difference, where the mycorrhizal hyphae, the fungal hyphae, are so much more petite, slender, and they can grow to massive extensions off the root. And so if you're out in the woods hiking, which I hope you did over the weekend, even though it's raining, the idea that as you're walking through the forest systems, every step you take, they've done calculations where it's mind-boggling the amount of kilometers of fungal hyphae underneath one footstep. It's just astounding. So they, of course, are invisible to us because we don't see what's going on in the soil on a day-to-day basis, but they are certainly there. And so we've been working through this material we have been focusing on the ectomycorrhizal fungi first, and some key characteristics with that mantle covering the root surface. The Hartig net is the growing of the hyphae in between the cells inside the root in the cell wall space. And then, of course, they grow off the root surface into the soil system. So we've been focusing on that. We talked about a couple of different kinds of ectomycorrhizal fungi. I showed you lots of really beautiful pictures of how they form that relationship on the root systems. Now we need to get into the endomycorrhizal fungi. So often the arbuscular mycorrhizal are just called endo, and then these are ecto, kind of a nice comparison, ectos and endos. But then some people refer to the endomycorrhizal fungi as arbuscular mycorrhizal fungi. Sometimes it's vesicular arbuscular mycorrhizal fungi. Some people shorten it down to VAM fungi because it's hard to keep saying vesicular arbuscular all the time. So let's jump forward now on the slide set to the endos for the VAM. Let's see, we need to go all the way down here. So I tend to say VAM rather than saying vesicular or arbuscular, but just be aware that depending on the person you're talking to or what you're reading, they may have different names for this. And I've seen more and more that people are just dropping the V and they're just calling them arbuscular mycorrhizal fungi. So in comparison to the ectos, they do not form a thick mantle of growth around the surface of the root. They are on the root. It's just very minor growth. It's not like a mantle. They're there, but it's easily, if you were to uproot the root system physically, like if you're repotting a plant, you would probably strip away much of the VAM fungal hyphae because it's so delicate and not very abundant growth on the surface. So less dense hyphae on the root surface. Certainly the hyphae still extend off the root. And again, when you uproot a plant, you break all those connections very easily. So often with the naked eye, you won't see VAM fungi like you can with the ectos, because that ectos forms that really thick mantle that you can see with the naked eye. But VAMs, they're tricky to see. They can penetrate past the cell wall. So they're a little bit more intimate with the plant root. So the ectos hang out in the cell wall space. So they're in between the cell walls of two adjacent cells. The VAM fungi can actually make it past the cell wall, and then they hug up or press up to the cell membrane of the plant cell. but they don't penetrate the plant cell membrane, okay? So that's, I probably should have picked something other than red. I'll switch that, but not the cell membrane. Okay? They do not penetrate the cell membrane. Okay, and I'm making a big deal about that because so many resources that you might read, they make it sound like these fungi are penetrating the cell membrane, and that's just not the case. Otherwise, if that did happen, the plant would kick off a defense response. So the reason they're called vesicular or buscular mycorrhizal fungi is that they form structures that are called vesicles, and they form structures that are called arbuscles. And vesicles are just balloon shapes. And arbuscle, if you look up that word, it means dwarf tree. I'll show you some pictures. So that's why they got that big long name. So when you look at the root system of a plant that has VAM fungi associated with it, we say it's not as extensive in terms of the biomass. Okay, if you were to weigh the VAM fungi associated with that plant root system, it rarely exceeds 10% of the root weight. So much more light growth of the fungal hyphae on the root. And the VAM fungi are really well adapted to hanging out with what we call herbaceous angiosperms, so the non-woody angiosperms. That doesn't mean they never form a relationship with woody angiosperms. It just means you see more species hanging out with the herbaceous. So when we say that, that's usually the one season plants with no wood or tree trunk or anything like that. It's just like, your corn, your peas and beans and wildflowers and things like that. Okay. All right. So looking at it visually, if you look at the root system, so the artist is trying to show here that the amount of fungal hyphae growing on the surface is minimal. You just see a little bit here. You see the hyphae coming off the root. The vesicles are being shown here. So it's just the fungal hyphae deciding to kind of balloon out a little bit into a balloon shape. It just increases surface area. They think that's why they do that. It helps with exchange of chemicals. And then here is the arbuscle idea. So it's all fungal hyphae, right? And it looks like a dwarf tree. And again, it's just repeated branching of the fungal hyphae. And again, the artist didn't do a very good job. It's really kind of hard to do it, but you have to imagine that the plant-derived membrane is surrounding all of this arbuscle. It's not just floating around free.
So the cool part, like, what it helps to kind of imagine is like a hand kind of holding this.
Yeah,
like
I know, right?
But it's like we can just think of it like holding.
Yes, or like if you slipped a hand into a latex glove, like the latex glove is the plant membrane, and your fingers are the fungal hyphae, if that helps. So there's always the plant-derived membrane around any part of the fungal hyphae.
Okay, so that can be inside the cell wall, but it's wrapped around. It's not.
Correct. The membrane is like wrapping around the fungal hyphae. I know. But I think if you think about fitting your hand into a latex glove, and the latex glove is the membrane. And so they're really hugged up to the membrane. It's a very intimate physical association between the arbuscular or the VAM fungi and the plant membranes. So they're, and then that allows for really good exchange of chemicals, nutrients, and all of that. So they think that the VAM fungi, if there's a lot of debate, but they think the VAM fungi came first, evolutionarily speaking, and then the ectos after that, but there's some debate still on that. Anyways, so here again, just showing some images. And so depending on the fungal species, they can look different. Some favor the arbuscle shape, some like vesicles, some do these weird coiling. It just depends on the species, how they look under the microscope. So with your experiments that you're doing in the labs, each lab section is going to be looking at mycorrhizal fungi impacts on plant growth. And hopefully at the end of that experiment, we'll be able to stain the roots. We'll collect some root samples and stain them, and we'll actually be looking for this. We'll be looking for arbuscles and the vesicles. And the staining method usually works quite well, where the fungal hyphae pops a very deep blue and the rest of the root tissue is rather colorless. So you can see it under just the normal light microscope. Here's a dramatic example of an arbuscle, and I mean, doesn't that look like a dwarf tree or a shrub? I mean, it really does. So you can see why they named it that. Unfortunately, it's just a mouthful to say the vesicular arbuscular all the time. Here's a root section with many root cells, and you can see how the arbuscles are clearly differentiated from the rest of the plant roots. Here's a close-up of another plant root where you're seeing the arbuscles and vesicles. So again, it's very easy to think that's floating around in the cytoplasm just by these images. And again, that's why I'm really emphasizing, no, they don't float around in the cytoplasm. They are always surrounded by a plant-derived membrane. Here's another couple images. So this is the staining technique that you'll be doing, where the stain will show the fungal hyphae with blue. The trypan blue stain will stick to the fungal hyphae and not to the root cells. And then just a close-up image on the top right here of that's an electron microscope picture of the arbuscle. So great surface area by that repeated branching like that really does allow for good surface area and good nutrient exchange. So again, the comparison of the two. So I'm a fan of compare and contrast on exam questions. So as you're looking at this material and studying this material, that's a good way to think about it. What's similar, what's different, compare and contrast. Okay, I think that helps you get a little bit more detailed focus on on a topic like this. So this relationship, they think, dates all the way back to when plants shifted from the ocean environment to terrestrial environments. So millions upon millions of years ago, they think the fungi were there to help that evolutionary process. That's a huge step in the evolutionary process. And they think, and I'm not going to go into detail, but they've actually looked at algae, and certain algae have the genes necessary to partner up with mycorrhizal fungi. So they're really starting to look at the evolutionary trajectory of the mycorrhizal fungi and the plants. And the current feeling and what the data is showing is that the mycorrhizal fungi were there right from that very crucial step of going from ocean to terrestrial environment. Okay, what else can we say? Well, the fact that so many of these plants that have been studied form this relationship. I said 80% on one of our previous slides. I see some experts quote 90%. There are some groups that don't seem to form this relationship or have lost the ability to do this, but it's very minimal. Most plants do this relationship. And so if so many different plants do this, there must be an advantage to this, because this is complicated. I mean, this is a symbiosis that it's very complicated in order to get this to happen. Lots of chemical communication. They have to find each other to begin with and then chemically communicate with each other. And just all of the gene expression patterns that have to shift, it's a big deal. And so there has to be advantages. So there's literally thousands of studies that have been published on this idea, and over they show that there is improved absorption of key nutrients, minerals that the plant needs. I put it in ranked order. Phosphorus is number 1, then nitrogen and potassium. So phosphorus in particular is very challenging for plants to access by themselves, but the fungus is super good at it. All right, and so they can get access to phosphorus that is also, the fungus can get access to phosphorus that has complexed out or precipitated out. The plant can't really touch that, but the fungus can, because they have the necessary enzymes to resolubilize these minerals. So it's a big deal. Water uptake, of course, just because the fungal hyphae can get into all the nooks and crannies of the soil that the fungal root just can't physically fit into. So great access to water. And over, these studies show, they compare mycorrhizal plants with mycorrhizae and then the control without, and over, the improvement in growth rate and overall biomass at the end of the season or the experiment is much greater in the presence of the mycorrhizal fungi. And then don't forget about protection, at least some, well, both BAM fungi and ectomycorrhizal fungi can do protection in the sense that both of them can give off chemicals to inhibit pathogens. So there's that biocontrol ability where they're excreting out chemicals that inhibit the growth of pathogens. That's one of the reasons I was so interested in studying them for my PhD work, because I was interested in their biocontrol ability. The ecto also, because they form that really thick mantle, they give physical protection too. All right, so it's just a nice physical barrier to potential either creepy crawlies that are crawling through the soil and want to try to munch on the root. That ectomycorrhizal mantle can help protect, or it might be another fungus, you know, a pathogen trying to attack, or a bacteria. So both physical and chemical are options for these mycorrhizal fungi, at least with the ecto, they can do both. So again, there are more and more businesses popping up that are selling these products, the mycorrhizal fungi. And so there's been some interesting studies that show that most of the companies that I've seen anyways, they tend to focus on the VAM fungi or the endomycorrhizal fungi, because those are the ones that form with most plants that people grow in their gardens. So for crop production, spam fungi are the best fit. Ecto, on the other hand, that's more like, well, maybe if you have an apple orchard, or, you know, limes, lemons, apples, pears, you know, ecto would be a good choice, and endo. So you, on one plant, you can have like a tree, you can have ectomycorrhizal fungi hanging out and the endomycorrhizal fungi. You can have both types. One of my friends who was a grad student at the time when I was, he did that successional study of mycorrhizal fungi in one ecosystem. He could show that there are mycorrhizal fungi that come in to, when you outplant seedlings, we were working with red pine. So we had a fresh site, we outplanted hundreds of seedlings, and then he tracked the mycorrhizal succession over time. And he could show that the species that first colonized the roots, that shifted and gave away to other species that would come in. And there was a definite succession going on over time in the types and species showing up. And so you can have one, like the red pine, we saw a lot of ecto on there, but there were also endos that were showing up. Here are just some images taken from different websites where they're promoting the sale of mycorrhizal amendments. I've used the Great White quite a bit in this class off and on over the years. That's one product, but there's many others since then. They were one of the earlier ones, but there's lots of other options. And they like to show pictures like this of, you know, with and without the mycorrhizal fungi. And so I just bought, for this semester, I bought a product. Some of you who might be familiar with these are MycoBloom. I bought that one. And then is it Mycos? So I bought two different versions. One bag is actually a mixture of, I think, 6 different the VAM fungi. And then the other bag is just one species, one type. And so that'll be interesting to see. So those products should work for this semester. So really what we're thinking about is this idea that when you have the plant roots in the soil, yes, the plant roots can take up water and minerals, but the presence of these fungi and other closely associated microbes, including bacteria, can be crucial for the plant to really thrive in that soil. And so it has become abundantly clear through lots of good research that the conventional agricultural soils, so much of that has been depleted in minerals, and because of the way the soil has been treated over the decades, it's been abused. And then also, so many of the fertilizers, the pesticides, the herbicides that are applied to those conventional agricultural field soils, so much of that chemical input actually kills a lot of the beneficial microbes. So they're actually using the phrase, it's a microbial desert The soil is really a microbial desert. And what they are trying to get across is that there's just so few microbes compared to a healthy soil system. And again, if you don't have good mixtures of the good, the right type of microbes, the plant will suffer because they won't get access to the minerals and then water and the nutrients. So I like this quote a company uses: we don't grow plants, we grow the soil. The soil grows the plant. And I think if you approach growing plants with that mindset, you're going to end up in a much better situation with a product, a plant that is thriving and giving you what you want from it. And so knowing, again, in my microbiology classes, I always tell the students, if you know a little bit of microbiology, you can go a long way, because you can apply so much of those concepts to all kinds of different disciplines. And certainly it's huge in the world of plants. If you want to work with plants for a living, you need to know some microbiology if you want to maximize your profit margin. So, and just have healthy plants in general. So looking at the mycorrhizal fungi, we're going to stop with the mycorrhizal fungi in terms of the microbes, but I could go on and on and on about all the other microbes that are there helping the plant as well. And I will touch on that a little bit later in the semester when we get into secondary metabolite production and how microbes can influence that process that the plants are doing quite dramatically. So, but I'll stop with regret. I'll stop talking about mycorrhizal fungi. It's one of my favorite topics. But now let's shift into a little bit more about these mineral nutrients that the fungi are helping the plant get. And so, thinking about the artificial fertilizers, the synthetic fertilizers, it's actually Dr. Liebig who is credited with being the kind of the instigator of all this artificial fertilizer. It actually, from my understanding and what I've read, it came out of World War II, where so many of the companies that were making chemicals to kill people on the other side of the war, once the war stopped, they're like, okay, now what do we kill? We can't keep making chemicals to kill people during the war, so let's make chemicals to kill plant pests. So that's what they should do.
They were areas that would have certain things.
Right.
What would we want over here that's making these plants?
Yeah, right. Yeah, I mean, just a simple observational experiment, or just an observation, and it led to this whole industry. So yeah, it's really interesting to learn about the history of this and how it fits into war and killing. But here, this quote is taken from a book, and it says, "What most don't know, is that later in his life, Liebig acknowledged a grave mistake in relying only on chemistry. In fact, he saw the negative impacts of artificial fertilizers on life in the soil in his own vegetable garden, and thereafter preferred organic matter to the inorganic." So the father of artificial fertilizers was like, oops. It has a role to play in some cases, but he was realizing before he died that organic matter was actually creating a healthier plant in his garden. So this is taken from the book called Teeming with Nutrients by Jeff Lowenfels. He's written several books. One is Teeming with Nutrients, Teeming with Microbes. There's at least one other one. Say that.
Teeming with fungus.
Yes, thank you. Of course, Teeming with Fungus. Can't forget them. So yes. Good. I've seen a couple of mistakes in his book, but overall very, very well written. So this green box is talking about Jeff Lowenfels, the author, and how, and I gave a seminar to Transition. Marquette holds like informational workshops for the community gardener people in the community. And I gave a talk about healthy, soil needs microbes, and the vast majority of people, they were just ordinary people living in Marquette that liked to garden. They had no clue. They were just like, what? They didn't know that microbes are so good for the soil and for the plants. And so it really struck home with me that this knowledge just isn't out there widely out there. And I was, I'm so immersed in it on a day-to-day basis as part of my research program. So I just live and breathe it. And so to realize that this is not really all that well known outside of these walls was a little bit stunning to me. But anyway, synthetic fertilizers, they had to figure out, what do plants need and in what ratios and how does this all work? So it was a lot of painstaking work initially. And really what they chose to work with was hydroponic systems, because that's much easier to control and manipulate than a soil system, of course. And so hydroponic research has been going on for decades and decades, because it was an easy way to grow plants, and you could put in your own mixture of these different minerals to find out what the plants need and in what amounts. And so many of you are very familiar with hydroponic systems, and if you're in our indoor ag program, you get exposed to many varieties of hydroponic systems that you can learn about with the nutrient film and the ebb and flow and the bucket systems. We have aquaponics going as well. So lots of options to learn about and play with and get experience on. And so with the hydroponic idea, it's some kind of liquid system typically, whether you have inert, sometimes the clay pebbles are in there too, but often it's just the hydroponic solution. You need to provide some kind of airflow so that because roots are alive, they need oxygen to do their respiration. So you have to provide enough oxygen for that. And then you have to make sure it's not stagnant water, it circulates, you have fresh nutrients coming in, waste products going out. That's the beauty of aquaponics, where you can use fish to clean up the waste, and then the plant gets purified water back to them. So, but basically, and depending on, I've done some work with tower hydroponic systems and aeroponic systems. Some of my research students are working on those. And we're actually looking at using organic amendments in hydroponics, which not much has been done with that. And I think that is a huge void of knowledge that we need to start picking away at, because I think there could be some really good applications for improving plant growth and actually tweaking what kind of secondary metabolites might be coming out of hydroponically grown plants with or without the presence of these microbes. So here you're seeing more of the aeroponic system. We mist the roots with, and then they're all over the droplet size. You got to get into that. Like what are the best misters? What is the preferred droplet size to maximize nutrient uptake? There's a lot to it. Ebb and flow. So thinking about, figuring out what plants need. Initially, all this research was done mostly in hydroponics, but you can also work with so-called soil-free physical substrates. And that is done. I'll mention a couple of those ideas. But with hydroponics, one of the key things is aeration. Those roots need to have oxygen to do respiration. And you don't want that in direct sunlight, because algae and cyanobacteria will tend to proliferate and overwhelm the system. And you need to know water chemistry, because you need to be able to do testing on it, make sure that you're not having selective ion depletion. So if the nitrogen runs low, you have to know that, and maybe everything else is fine, but it's just low in nitrogen, and so then you have to tweak the recipe to make sure that all the ions are getting into the hydroponic so the plant has access to it. And then pH changes. You don't want it coming. What's the pH range most plants like? Close. 5.5 to 6.5. 5.5 to 6.5. So same in hydroponics in general, they like that. And then I've seen this quote, and I would like you to say, no, not necessarily, because I kept getting told this over and over. Mycorrhizal do not form in hydroponics, but there have been a couple of key studies where they show that mycorrhizal fungi are there and they persist.
Where do they come from?
Well, in these controlled studies, they were spiked in. So this was a controlled lab-based thing. I can dig out, if anyone's interested, I can dig out at least one of the papers I know I just saw the other day in my PDF file. So So that is a big question mark. I think that's something, and in fact, one of my grad students did end up spiking in a mycorrhizal fungus to see what would happen. And then part of her work was looking at what microbes are there at the end of the experiment, because putting them in, that doesn't, they might have all died like that. You have to see if they're present and persisting. And so she was taking samples throughout the growth and at the end. So we'll see. She hasn't crunched the data yet, so I don't know. So sometimes, and if you think about it, you could see where a physical substrate might be good for the roots to grow into, because that's how roots evolve. They evolve to grow through soil particles and around soil particles. And so you can see where that would be a little bit more natural to the plant. So sometimes they will use like acid washed quartz sand, just means they've washed any minerals off the surface of the sand particles, so it's inert, but it's a physical substrate that the roots can grow through. Perlite is another inert substance. It's volcanic rock that's been heated and treated, and it's that white poofy stuff, kind of looks like little popcorn bits, and that is perlite. And then there's vermiculite. Vermiculite is another inert. And so you often see perlite and vermiculite added into potting soil because it creates loft so that your soil doesn't get too compacted. But it's also inert. You could use like pure perlite, like we're growing kidney bean seeds for you right now in perlite, because we're going to transfer those beans into a hydroponic system. And so we don't want soil coming with that. And so we're growing them in perlite, and it doesn't really matter if some perlite bits get into the hydroponic because it's inert and it won't mess with the experiment. All right, so there are ways to grow them in inert physical substrates if you want to do that. So all of this research, what they came down to is they said, yeah, plants have essential nutrients like any living organism. And so they define that as in the absence of that nutrient, they're not able to complete their life cycle. That makes sense if you think about it. So another way to think about it, like, oh, the plant might be able to grow, but it wouldn't make it all the way to flower and seed production. Or maybe it could flower, but not do the seed. And so somewhere along the line, it just peters out and dies out. And then the second part to that definition is that the mineral is a key part of some molecule that the plant needs in order to function. So a classic example of that is chlorophyll. The chlorophyll pigment actually has to have magnesium chelated in the pigment. And we'll get into that when we get into the light reactions. So magnesium is incredibly important for plants. And unfortunately, so much of our conventional egg soil is highly deficient in magnesium. So much so that that's one supplement I take on a regular basis, because I just don't feel my diet is providing. Did you know that your ATP has to complex with magnesium to fully function? Do you know how important ATP is to your body? And if you don't have magnesium, it's like a big deal. And once I didn't find that out till just a handful of years ago. It's like, how come I don't remember learning that in physiology class? But if you look it up, you have to have ATP has to be, I don't know if it's chelated, but bonded with magnesium for it to be fully functional. So that is one. Oh, and by the way, speaking of magnesium supplements, if you go to Walmart or whatever and get like magnesium oxide, it's really cheap. There's a reason it's really cheap. She just essentially, it goes right through you. It's not bioavailable. That's a whole nother ball of wax. You got to look at the bioavailability of any of these minerals that you might want in your diet. So be really careful about that. I've done a lot of research on magnesium, and it's just so irritating what companies will put out, and they'll lie to you basically and say, oh yeah, this is good for you. Well, yeah, maybe if you're constipated because it'll make you poop, but it won't do anything else for you. So be careful about supplements. It's the wild, wild west when it comes to stuff like that, unfortunately.
The two key things that
Yeah, so this is like the two-part definition of what an essential nutrient is. Okay, so macronutrients. Macro just means they're present in large amounts. And what that usually means is that they are part of a structural aspect of the plant body. And that's why they're needed in such large amounts. So macronutrients, and then there is debate on some of the micronutrients we'll talk about. Some people think, especially like iron, maybe that should be considered a macro. The debate still rages. But micronutrients, they're required in small amounts, but they're still important. Just because they're called micro doesn't mean they're unimportant. It just means they're required in small amounts, but they're absolutely essential. So that means they usually form a catalytic and regulatory role. So they are important in some physiological pathway. They're necessary for enzymes to fully function, for example. Okay. So the plant physiologists did all this hydroponic study, and basically what they were doing is they create a mineral solution with one of the nutrients held out of it, and then grow the plants and see what happens. And that's actually what you're going to do in lab, in the mineral deficiency lab. We're going to set up different solutions that have different things missing from it, different minerals, and you'll get to see the impact on how the plant looks while growing in that deficient solution. And so they usually exhibit key deficiency symptoms that you can look for and train your eye to as you work with plants. So here you're looking at, of course, all organisms need carbon, hydrogen, oxygen, but here we're looking at what's being taken from the soil, the macro versus the micro. And you can see that there's quite a list. And what they do is they put it in relationship to molybdenum, which is really hard to say. So this last column is saying that you need 1 million times more nitrogen than you do molybdenum. Okay. So let's look at this. So this next table is the one I want you to study. So this table, 4.2. I think it's still 4.2. Huh?
No, she doesn't.
Oh, table like 85, table 85, what? Table 4.2. So you'll see that this is grouped, group 1, 2, 3, 4, and we have the whole statement here. That's what I want you to know. So I want you to put these minerals in relationship to what they do for the plant. And so, understanding that nitrogen and sulfur are part of the carbon compound, and then they give you some excellent examples to get you thinking about it. Group 2, phosphorus, silicon, and boron. They're necessary for energy storage, structural integrity. So these bolded phrases are important. And then knowing some examples. So if I gave you like a matching or something, could you pick out, for example, that? So if I gave you a list of, and Cassie was on it, would you know that that belongs to the group that's in ionic form in the plant? Whereas if I gave you iron, would you know that that's involved in redox reaction? So that's how you study that, looking at what these minerals do for the plant, rather than just a list of macro and micro. I want you to start thinking about what they do for the plant. That's important information. This next table is just looking at a Hoagland. So this is named after Dr. Hoagland, who came up with a standard hydroponic recipe that's used by millions and millions of people, and there's all kinds of modifications of it. So I'm not going to ask anything directly about this. It's just for your own knowledge. We will be working with a modified Hoagland solution in lab when we do the mineral deficiency. Okay, so that's just for your own use. Okay, a couple of ideas. So when you're looking at a plant, and people have come knocking on my door and they say, "What's wrong with my plant?" And they either show me a picture or they have it with them. And so if I'm going to try to help them figure out what's wrong with their plant, of course, first you have to figure out, is it a plant pathogen that's doing this, or is it a mineral deficiency that's doing this? If it's a deficiency, one thing you can look at is the whole plant body. You look at the younger parts of the body versus the older parts of the body. So usually, of course, you only see the above ground parts. So you look at the leaves. The leaves that are closer to the tip of the plant, top of the plant, those are younger. The older leaves are at the base of the stem. So look at those two, and that will tell you if a deficiency is a mobile or immobile element causing that deficiency. So what we mean by that is if the mineral is mobile within the plant, it can get redistributed. So when the plant takes up the water and the dissolved minerals, it can send that mobile nutrient. So let's say the nutrient ends up in some mature leaves, and then some young leaves sprout, and they go, I need more potassium than what you're giving me. The mature leaves can say, I'll give you some of mine. And they will mobilize that potassium up to the younger leaf. And so that's what we mean. It's like the older parts can sacrifice and say, I'll give you some. All right. Well, that means that if that keeps happening over and over and over, you can imagine where the mature leaves will then exhibit symptoms of deficiency first because they're sacrificing all of that mobile element up to the younger leaves. And so you'll see that pattern where the old leaves look disease, something's wrong with them. The young leaves still look okay. So look for that. That's a mobile element causing that. Immobile, on the other hand, that's where once the element is deposited in that whatever part of the plant body, that's it. It doesn't get mobilized. So even if a younger active growing part of the body says, hey, I need that, the plant part that has that mineral says, oh, well, so sorry, I'm hanging on to it. I can't get it to you. All right. So immobile, once located, it will not move. So that means younger parts of the plant body will start to display symptoms first. The older ones will still look fine. So that right there can give you a lot of information. Just looking at the entire plant body, young versus old, parts of the body. Here, I would like you to know this table, and the way I would suggest studying it is if you memorize what is immobile, then all the rest is mobile. All right, and that can vary depending on the expert who's saying this. So I've seen tables that vary. In fact, I might have some in here. So wasn't there a show that was named CSI for a while? I remember a student popping saying, "No, that's CSI. That's easy to remember." And so there's little acronyms and ways you can memorize that. But these are famous for being immobile, calcium, sulfur, iron, boron, and copper. This next one here is, let's see, this is, wait a minute, no. I know. So here I put zinc in parentheses because I've seen that in the immobile category, even though our book is saying it's mobile. So there's a little bit of confusion here. This is a different source. I think this is from Cornell University, and this is kind of nice because it shows a little bit more information about them, but they're showing actually the mobility in the plant as well as in the soil. So zinc, they're claiming from Cornell that, zinc is immobile, but I've seen other resources that say it's mobile. Take it with a little bit of a grain of salt on that. So if you're good with this one, I think this is the one from our book. So this is the one I want you to know. Okay, so mobile versus immobile, we're talking within the plant bodies, looking at deficiency symptoms. Okay, so what are some terms that you would use? So if I'm looking at a plant and I say, oh, the young parts, the young leaves are showing chlorosis. What that means is it's yellowing. Okay, you get that yellow look to the tissue. And if I say, oh, it's displaying general chlorosis, that means the whole leaf, for example, the whole leaf, the entire leaf is going yellow. It's just kind of even yellow. Then I might say, oh, that leaf over there is showing mottled chlorosis. That's spots, dots, spots of chlorosis. So it's a good portion of the leaf is green, but you get dots or patches of yellow. And then I might say interveinal. That means that the veins look distinctively green still, and the tissue just flanking the vein is green, but the rest of the leaf is yellowish. And I'll show you a couple of pictures to show this. There's also the term necrosis or necrotic. So chlorosis is trying to say a lack of chlorophyll. So it's no longer green, it's yellow. Necrosis means it's taken beyond that and you're going to brown and black tissue, dead. It's like dead, dead, dead. It's never going to come back. All right, so necrosis is dead tissue. Chlorotic tissue can come back. If you feed it the right stuff, it could green up again. All right, it's possible. So here are some visuals, just trying to show kind of general chlorosis, mottled chlorosis. I think I have a better picture of interveinal. So this is just showing some ideas and different, magnesium, for example, the lower leaves turn yellow. Veins remain green. That's not a very good interveinal, but here, let me show you some other pictures. So here's general chlorosis, evenly yellow, very good modeled chlorosis on the right there. Here's a really good interveinal. Oh, yeah, that's true. I mean, I've seen healthy leaves. This isn't, it's just the pigment pattern for that particular species. So you do have to be careful. Yeah, you can get, and they're fine, they're healthy. So start training your eye. If you have houseplants, you can start telling, you can tell a lot about what's going on. If you have deprived that plant of certain key minerals, they'll start to let you know. They can't scream at you. It's a good thing plants can't talk. I'd have a very noisy house because very often I'm sure they'd be screaming at me if they could, because I forget sometimes. All right, so the 3 numbers. So if you've gone to purchase fertilizer, you typically see the NPK ratio on that bottle or bag of fertilizer. So the nitrogen, phosphorus, and potassium. And you can buy different fertilized mixes with different ratios of these 3 minerals. So these are the big 3, but remember that's just 3 of the macronutrients. There are other macro and micronutrients. So, and it's always by convention done in the NPK. The numbers are laid out that way. Okay, so NPK. That's how it's always done, no matter what bottle you look at. Okay. So here you can see nitrogen. Typically, you need that for really good shoot and leaf growth. Phosphorus is good for blooming and flowering, and potassium is just good all the time, pretty much. Potassium is actually used pretty heavily by plants as well. Okay, so we're going to look at some of the macronutrients. I'm not going to go through all of them. So some of the material on the slides I won't directly talk about, but you're still responsible for what's in the slide set. And then same with the micronutrients. I'll mention a few key things, but there's other slides that you need to keep up on. Okay, so make sure you're studying these slide sets. All right, we'll stop there and continue on Wednesday.
You mentioned like the father of artificial fertilizer.